My guest today is Bert Hubert. He is a software engineer by day, geneticist by night. His work in genetics has been published in Nature, specifically in their multidisciplinary Journal of Microbial Ecology. He took interest in genetics because of the parallels he noticed between the genetic code and assembly code in computing. In fact, he was so blown away by the parallels that he started a website, this was years ago, pointing out all the similarities. This ended up leading him to start conversations with geneticists, and eventually, this blossomed into him doing actual scientific research that's published in Nature. Our discussion today was pre-recorded. We weren't able to match our schedules to do a live show. But in it, we talk about many things, including his encounters with creationists who think that his work, pointing out the similarities between computers and cells, counts as evidence of God. Bert, of course, does not take that creationist view. In our chat, we focus a little bit on what's known about the evolutionary origin of the genetic code, but for the most part, we just focus on how amazing it really is. Just think about this for a second. At the very heart of biology exists a true communication system. Genes transmit messages, which are received and decoded by ribosomes. Genes are molecules, ribosomes are molecules. This is insane. Molecules, they have no minds, they have no understanding of their own existence, yet they have evolved the ability to communicate. Scientists have discovered the foundational rules of their strange language. You can see those rules laid out in the codon chart of the standard genetic code. These rules are nearly universal. Almost every single organism uses actually this exact same genetic code. There are some slight variations. Many more rules that govern gene regulation have been discovered on top of this, and these are oftentimes species-specific rules, and many more rules still wait to be discovered. So hopefully today you've got an appetite for curiosity as I introduce you to this fascinating man who goes by Bert. Bert Hubert, welcome to Sunday Morning Science. Bert, can you tell us about your background briefly in computers and then the actual research and the work that you did in biology?
I studied physics originally, and I really loved that. And that was the time when the internet was really booming, and 1997. And every bad idea got founded as a company back then, and even my bad idea got founded as a company back then. And that's how I got into sort of running software, writing software that runs the internet. These days, around half of the internet in Europe and maybe 40% worldwide runs through software that I wrote. It's called powerdns.com. Yeah. And you're not obviously aware, but if you buy a domain name somewhere, chances are that it's powered by my software.
Oh yeah, yeah.
And it has been moderately successful, as they say. And but for a while it was also not that successful, which meant I suddenly had a lot of time on my hands. And sort of back in 2001, I ran into DNA and I read how these biologists were talking about it. And I thought, well, this is very fascinating stuff. And obviously these people are brilliant and they're finding out many things. But I found that it made sense to sort of apply my computer language and world and study what they were doing and see if it maybe also made sense to look at it that way.
Yeah, and you built a website about this, about looking at DNA from a programmer's perspective, and you did 2 videos, which is how I was introduced to your work.
Yeah, the original website, which is still online, is called Amazing DNA, and I wrote that, and that was just my first observations when I was reading through the big DNA textbooks, and I was like, hey. These people, they talk about codons, and we'll get to that later, what they are. But they're just like bytes, and they have bits, and these bits, they make up those codons. And so I assembled all the cool things I could find out about DNA so from a computer programmer's perspective, and I called that the Amazing DNA page. And it actually stems from 2001, so it's like, it's old by now. But it did attract serious attention over the years because I worded it carefully. I said, look, DNA is not a computer language. It is not a computer, but it still makes sense to reason about it like that. Mm-hmm. And over the years, many very serious DNA scientists would chime in and say, yeah, well, you got this sentence a little bit wrong. Yeah. And this needs a little bit of tuning, or I have this even more amazing story that you should put in there. So over the years, I finally sort of came to realize that, maybe I am onto something. Maybe this is a valid way of looking at DNA.
I actually first heard about you from a creationist who's claiming that your work proves that God exists because the cell is like a computer.
I get a lot of email like that.
Yeah,
The webpage and the two videos. So in the end, the two videos came from a hacker conference, and that was sort of honestly a sort of a high point of my sort of scientific life, or whatever you would call it. But there was like, I think 2,000 people listening to a story about DNA.
Yeah,
and that's good stuff. And we overflowed the time, and we had an extra session where lots more people showed up. So people do really care. But on average, I get like one email a month from someone that sees the hand of God in DNA, or Mars, or Atlantis. There are many people that sort of see things in DNA, and they all send me email.
Yeah, I did a video last week on signaling theory, which is the theory in biology that explains how it is that signaling systems, communication systems develop naturally. A lot of people are not familiar with that. And so when they see that there is something that we can call code, we really can call a genetic code a code. It really does work like a computer code. That the parallels are insane there. When people see that and they don't understand signaling theory, they think, oh, well, this must be some sort of intervention by either an alien or a intelligent designer and so on. And so I've been teaching evolution now since 2012 online. All my energy goes to online, science education. And this has been a theme in the comment sections of my videos and emails that people send to me, is that because the genetic code is a code, it had to have been designed, therefore aliens or God or whatever the person happens to believe in. And a lot of the online atheist community has been in the comments in my videos too, telling them, no, the genetic code has nothing to do with a code. It's just chemistry. The codon chart is just the same as the periodic table. It's just a chart showing molecules. And that's not true. And so I've been seeing these people argue back and forth, and it's just been, every once in a while I've tried to intervene. There was a atheist YouTube channel that does like a weekly show, and you can call in, and they had just been making fun of a guy for saying that the genetic code actually works like real code. And so I called in and tried to correct them, and they would not have it. They'd shut me down. They ended up like, so it's really weird that there's, I mean, I'm not religious. I don't believe that aliens or gods invented the genetic code. It's really weird to see that the atheist community, there's this, obviously it's not the whole atheist community, but there's this chunk of people in the atheist community that are just trying to throw away the significance of the genetic code in order to win an argument, or to pretend that they're winning an argument. And it's unfortunate, because the genetic code is so fascinating. What cells are doing.
It's utterly awesome. I mean, it's just, if you're seeking, if you look into it and you study it from time to time, you just go like, yeah, this is just the most amazing thing ever.
Yeah. Yeah.
And then you eat a banana, and then you realize, I'm eating this masterwork.
Yeah.
And it is just literally awesome in the sense that it's full of, it generates awe.
Right.
And many people have, and I think that's the reason why lots of people latch onto it also, is that it does instill that sense of wonder. Mm-hmm. And I have compared it in the past that let's say you live in a house, and suddenly an alien spaceship would land in your garden. Mm-hmm. And aliens would walk out, and they would offer you the engineering manual of their spaceship.
Yeah.
And that would be on the same level of fascinating as the DNA stuff we're discovering right now.
Right.
Because it is machinery. It powers life. It powers everything. It takes decisions. It is us, but it's not built by us.
Right.
So we see organisms make choices. We even see bacteria deploy strategies. Mm-hmm. And we don't really know how they do it. And then we can go look in the DNA, and there we find, your survival strategy is encoded in this glorious machinery.
Yeah.
Which we have not built.
Yeah.
It was built by nature, and there's for me, there's no reason to involve any religion or stuff in that. But it's awesome enough that we have these problem-solving algorithms that we did not make.
Yeah. Yeah.
And that by and large, we also do not understand. Because we can apply a tremendous amount of research, and then we understand a very small bit of it. and that's awesome enough. But we already know that so every time we discover something as human beings, like a control theory or whatever, we find out that it has been in nature for 300 million years already.
Yeah.
Everything we're going to discover in the coming decades is also already hiding out in there.
When we invented pulleys and levers, that's already
Yeah.
in our arms and hands and fingers. I mean, that stuff's already there. Pretty much everything that we've invented has been there before. I mean,
and in spades. So
Yeah.
the classic control theory, for example, is when you have a big ship and it needs to steer itself to stay on a certain heading.
mhm.
And if you're a very nervous guy, you would constantly be steering to keep the ship exactly on the right heading. But that's expensive because it costs fuel, it costs attention, but it was a task that human beings were uniquely good at. There was a very long time it was not possible to automate this.
Right.
And then finally, someone derived the math to actually keep a ship on the right heading with minimal interventions, and they were some nice differential equations. And then, like decades later, we find that the pH levels in our blood are governed by the exact same equations and the exact same mechanisms.
Yeah.
But it has been there for like 200 million years.
Yeah.
And so yeah, it's just it's full of awesome. There's no other way to go about it.
Yeah. So today we're not going to really talk about origins in general of this stuff. I've talked a little bit about that in other videos. I just want us to get just awestruck by just the beauty of these systems, and really try and understand from a coder's perspective, how this stuff relates to the computer. Because I mean, it's an analogy to computers and cells, but the analogy runs so deep that it's pretty spectacular. And one of the things that I just wanted to start with is, what is it that people really mean when they talk about computer code, when they talk about code in computers? The genetic code, it was called the code because that was in reference to the codes that people would use in wartime to communicate with each other secretly. And so when we cracked the genetic code, it was like cracking the Germans' code they were using in World War II. But what do we mean when we talk about code in computers?
It exists on multiple levels. So if we start down with the lowest level in a computer, there is a central processing unit, a CPU.
Yeah.
And that's the lowest level thing there is. And also that's the closest part to DNA that we actually see. And it executes code. And the code it executes is of the type, take these two numbers, add them together. Mm-hmm. And if the result is bigger than 100, then jump to this further bit of code and then execute that.
Okay.
So everything you do boils down to such simple to execute code that really does nothing more than adding numbers, comparing numbers, multiplying numbers, storing them, and maybe doing something in case a certain condition is met.
Now, is this, when we talk about ASCII code, that zeros and ones, is that what?
Yeah, this is actually even lower than ASCII code.
Okay.
These are really the zeros and ones. So if you really drill down to it, then a CPU takes like a collection of ones and zeros, and based on 16 of these ones and zeros, it does something, or it does not do something. And this is the most basic, and that's not the right word because that's actually a programming language, but it's the most fundamental code we have in a computer. And if you go back to sort of the original chips, so if you used to have a Commodore 64, for example, in the past, there was a chip in there with 3,000 transistors, which is a sort of manageable number. You could have them, which are little electronics units that are like this big. They're really tiny.
Yeah.
And you could have 3,000 of them, and those would already execute this code. And that's the most basic stuff that we have. And if you look at people that write JavaScript code, that is also code, but it is actually converted into these ones and zeros before it can actually run.
Is there any human that can read that stuff?
Yeah, and there are people, and there are worrying people. These are the scary hackers. These are the people you see on TV that hack into the Pentagon or whatever. Those are the people that can just see those ones and zeros, and they can not only figure out what is going on, but they can even see, well, this was originally written in the following computer programming language, and It was probably compiled in 2005 because there's a sort of handwriting in there where you can see, I can see where this came from and what it is doing.
Yeah.
So that's really, really fascinating stuff. And the same goes for DNA. If you look at the raw DNA, which is it's a fun thing to do, you can just download DNA and then you will read it as 4 letters, A, C, G, and T. So where computers have the zeros and ones, DNA has A, C, G, and T. And much like if you look at the zeros and ones, it takes you an awful long time to figure out what's going on. You can stare at DNA all day long and try to find meaning in there. And sometimes you can. Sometimes for example, there are people that are so good, they can take a look at DNA and say, well, this came from an E. coli-like bacterium. Right. Because I spot patterns in there that are typical of an E. coli bacterium. Right. Where it is utterly fascinating that the DNA of bacteria or viruses or us or plants and cucumbers are the same.
And what you're saying by that is there's not variation in the rules of the coding system. So the codon chart.
Yeah, it's to the point that code will in fact accidentally run.
Yeah.
So if a bacterium gets lost in the wrong place, then its DNA code will get executed, will get run, will have effect on human beings. And
there are a few tiny variations
but those only actually make it only more interesting.
Yeah.
Because and we'll get to that in spades, but the variations are so tiny and they all have reasons. They all have reasons where we can say, I can see why that happened.
The physical substrate that the genetic code runs on top of is the ribosome. It's the messenger RNA. It's this whole translation system. All of this is physical. You've got chemical reactions happening. These are evolved structures. The rules governing the genetic code emerged from these evolved structures. Sometimes people get this idea that code is somehow different, or that information is somehow like this magical thing that's different than normal matter. But of course, a computer actually runs, like all the information is stored in a physical substrate. Could you talk a bit about that physical substrate in the computer? What is actually happening? What are these zeros and ones really?
Yeah, well, actually, so the analogy is quite good again. Yeah. So in computers, actually, the ones and zeros, they sort of travel and they appear in different guises. Mm-hmm. So if they are on a rotating, spinning hard disk, which we all used to have, and there the information resides in transitions in magnetic poles between north and south poles. Okay. So a zero is actually a transition between a north and a south pole part of a disk. And but if it's in flash memory, then your one is a bunch of 20 to 40 electrons. Okay. That sits there and is trapped there and it cannot leave. That's why flash memory can work without power and retain your data for months. And actually, we don't even quite know why the electrons don't escape from there. But we're not asking because it works. If it's in your computer memory, then it's the same thing. It's a charge, the 1 and the 0, but the charge would leak out in a very short time. Yeah. So there, because it leaks out all the time, we have to keep on putting it back in. So many times per second, all your RAM memory gets visited. See, is there a charge in here? If so, let's top it up. Yeah. And if there is no charge in here, then let's make sure that it's empty. And so depending on where in the computer your information is, it is represented in different ways. By the time it goes to the computer chip, it's purely a voltage. Yeah. And it's like a mains voltage or not. And it's a few volt, which doesn't sound like a lot. But because chips are so small these days, a small voltage over a few nanometers exerts more charge differential than a bolt of lightning. So it's actually very impressive that they're able to process these bits and bytes through computers that are that small without them exploding. So that's the physical nature of it. And I try to see if you could make a parallel into the efficiency of DNA. And it's actually sort of quite doable. Sort of similar amounts of matter are involved in storing DNA as in the physical memory, except that DNA is wound extremely tightly. You could store petabytes of data in DNA in in the tiniest drop of water, and it would also be pretty stable. So DNA is really impressive. But in all, the analogies are quite good. A 1 and a 0 is either a bunch of electrons or no electrons, or a magnetic charge. And if you look at DNA, it's one of 4 molecules, which we call A, C, G, and T, which are quite tiny molecules. And we can recognize them by the different charge profiles that they have, which is, again, refers to electrons.
Information is not some woo-woo other dimensional thing. It is stored in matter. So in a computer, this is through electric pulses, or these atoms inside of these disks, these polar charges. And DNA, it's stored in molecules.
I can take out the magic further. We can now fully read DNA and just store it on disk. And if you then look at the file, you can literally have a sort of chromosome directory that has all these human chromosomes in there. If you then take a listing of that file and sort it on order, you find that our chromosomes are actually the numbers we assign to them, which is based on their sizes. So the fact that chromosome 1 is chromosome 1 is not because it's in any way more important, but just because it's big.
Right.
But we can now go full circle. You can read DNA from nature, store it on disk as a file, which you can just inspect. And then you can send back that file to a lab, and they will turn that file back into DNA again.
Yeah.
Which is sort of the ultimate way of taking the magic out of things and saying, I could read it to you over the phone. As a signal, as a voice signal, and then people would put it in the DNA synthesizer, and exactly the same thing would come out. So there's no magic in there, and people get all hung up about entropy and other stuff, and there's no more magic in a piece of DNA than there is in a flash drive.
Right.
And I consider both to be pretty magical in other ways, but it's not any woo-woo kind of magic. Right. It's very advanced technology.
The physical substrate that DNA is running on, it's the ribosome and the transfer RNA, and there's a really neat bit of research done on the ribosome. It has not the core of the ribosome. There's this tube inside the ribosome where the proteins come out as they're being coded, as they're being built. That has not changed the width of an atom in all of life. So since the ribosome came to be, it has not changed by a single atom in that little core. There are lots of things. If you look at a human ribosome versus a bacterial ribosome, there's lots of differences on the outside. There's lots of differences in how it attaches to messenger RNA and feeds that messenger RNA through it. But at its very core, it's atom to atom identical, which is spectacular. There's nothing like that aside from the ribosome in evolution that has stayed so stable.
But it has a good reason. You can actually compare that a little bit to computing code. There are mistakes in computer science that we made 30 years ago, and we're still dragging along those mistakes. Because by now, if we want to fix them now, then it's going to be terrible, because we would have to change everything at the same time. And since we're not going to change everything at the same time, because there's no way to coordinate that, we're not ever going to change it. And there are parts of DNA that are just like that. That if you want to change something really fundamental, then the whole organism would have to change synchronously. DNA that used to work on Monday would not work on Friday.
Right.
And that change is not going to happen. And because it's not going to happen, nothing has changed there.
Yeah.
This is a very strong analogy to the computing world, where you just see, it sucks the way we built it. Sorry for that. And we have to live with it now.
Yeah.
We have not only to live with it, we are it now.
Right. If you were to do any major change to the genetic code, it would break every single gene in an organism's body. So you would have to edit every single protein.
And yet we do see some, and I don't want to delve too much into the origins, but if you look at sort of the fingerprints of DNA and the way the DNA letters correspond to the amino acids and the proteins, you can sort of derive that right now we take 3 DNA letters and turn that into 1 amino acid. Yeah. But you can see that originally it was probably 2 amino acids. Mm-hmm. And one day life apparently decided that 2 was not quite enough and we needed 3. And life found some way to wiggle itself into 3. But from the architecture, we can make a good case that there are remnants where you can say, well, originally it was 2. And how that happened, you can reason about it, but that was sort of the biggest architectural change in life that ever happened.
Yeah, and that would have happened before the most recent common ancestor of all life, because all living things share the same genetic code. We can be very, very confident that we all share a common ancestor that was using that code.
Yeah.
And there's there's arguments to be made that we should think of the common ancestor really as the ribosome. We don't know if life was cellular at that time yet. We don't know a whole lot about what was going on. Maybe there was so much horizontal gene transfer that there were cell-like packages, and these genes were getting in to, infecting other cell-like compartments. And so that ribosome was the translation system.
Yep.
Was the thing that really took over and is our most recent common ancestor.
It is, and it is the most sort of craziest piece. So it does the conversion. It takes these 3 genetic letters and turns them into an amino acid, and that's the most fundamental thing there is. And even though it is more or less the same in the 3 domains of life that still exist. Mm-hmm. And indeed, we know that they must have been present in the 2 original domains of life, and yet yet it's mostly identical.
Just for clarity, for people listening, he said that the codon makes an amino acid. It doesn't actually turn into an amino acid. It's read and uses the ribosome uses that to know which amino acid to grab out of solution and add to the growing protein. So this is really it's a reading system. The messenger RNA with these codons on it, these 3 nucleotides, that's being read by the ribosome, and it uses that to know what to add to the growing protein next.
The order picker.
Yeah.
So it gets a list. Yeah. So you need to take this amino acid, you need to take that amino acid, and put them all together, and then you get a protein.
And this is why I'm so adamant about saying that this is a communication system, because you have a set of symbols. The codon is not an amino acid. It represents an amino acid. It is a symbol that was selected. These codons that we have in the codon chart, the classic codon chart of the genetic code that was cracked back in I think it was the 70s that they got the Nobel Prize for cracking that code. This was selected by nature to be the genetic code, and it could have been alternatives instead. This really is a communication system.
So there are 3 codons that correspond to 1 amino acid, and because there are 4 nucleotides, you will get 4 times 4 times 4 is 64 possibilities.
Yeah.
But there are only like 20 amino acids.
Right.
Which means there are more ways of saying the same thing in DNA. And anytime you get that kind of freedom, there are more ways of saying the same thing, you can get the possibility for dialects. And what we find in nature is that different strands of nature have different preferred ways of encoding the same amino acid. So some of them will always use AAC to describe alanine or whatever, and some of them will use AAG. Right. And those are dialects. And those dialects, they do not break the compatibility in any way. But for example, when the insulin hormone was first made using DNA, because diabetic people need that, and they took the human genetic code for insulin and put that in a bacterium and told them to go run with it. And the bacterium dutifully did that, and it built insulin, and everyone was like, yay, Nobel Prize, actual Nobel Prize.
Yeah.
But it didn't, the bacterium was not in any hurry to do that. It was actually very slow. It really was like, people were disappointed. It was not making a lot of insulin.
Yeah.
Which is, of course, nice. You have this revolution, and then 3 weeks later, people say, yeah, well, it's a bit slow. And then they looked at the codon tables for bacteria and for human beings, and they found out that actually we were using valid DNA for the bacterium, but for the bacterium, it was like we were speaking a very heavy dialect.
Right.
Because it was using codons that are frequently used in humans, but very infrequently in bacteria.
Yeah.
And then the code was adjusted to the bacterial dialect, and it went like 3 orders of magnitude faster.
Oh, wow. Yeah, I did not
Which
know
is
that.
the equivalent of 2 people both speaking English, but one with a very heavy accent.
Right.
And yeah, they can still understand each other, but it's going to take more time.
Yeah.
And then you adjust a few things, and then you're like, oh, that's what you were saying.
Right.
I find this utterly fascinating again, of course. But then we find out that this dialect is actually also used for a lot of things.
Yeah.
Because we now have discussed how you take a bunch of DNA with codons and that converts into amino acids and in proteins. It's nice if you have a protein factory, but a protein factory is not yet life. We need to know when to make what protein.
Right.
And that is where you get to the core of what the overlap between computer programming and DNA, because you want to have algorithms.
Right.
Where you can have if statements that say, if there is a lack of glucose, then make the following protein. Right. Or if there is too much glucose, then produce insulin. And those, it took a long, this is where the biologists are really, it's sort of, I love my biologists because many of them will be watching this. I love my biology friends. I really do. But when I see them write about algorithms in DNA, it's sort of sad to see. Because they're this, it's like someone that cannot cook who is describing how to cook an egg.
Yeah.
And they really and whereas we as computer people, we go like, yeah, I see an if statement there.
Right.
And where we can see pieces of DNA that are turned on under certain conditions. And here's where it sort of gets magical. DNA needs to get activated under certain conditions, and there are many ways of doing that. But to do that, the DNA itself needs to have a certain charge distribution. It needs to have a certain physical shape. But if you look at purely the code itself, and let's say every codon corresponded to one amino acid, you would not have any freedom to shape DNA in any way. The DNA would always be the same. It would be only one way to describe insulin in DNA. But because we have so many different ways of encoding the same amino acids, you can maintain the amino acids of insulin, but change the DNA such that it will curl up in a very interesting shape.
Right.
And we can do that because of these dialects. And there you see a very big difference between computer code and DNA code. When someone programs a computer, it doesn't matter if they add spaces to it or empty lines or whatever. The code is the code. But for DNA, actually, the way it is written very much determines how it will be run.
Yeah, so
there is metadata in the signal.
So you're saying that the sequence in a genome, not only is that used by the ribosome to build protein, but it's also used by packaging proteins inside the nucleus of the cell, in our case, or just inside the bacteria, to wrap up that DNA and package it in different ways.
Yeah, so that it will get activated, or so that it will rarely get activated, or so the shape of the DNA determines when it will get activated, and then the contents of the DNA will determine what happens then. Yeah. What protein comes out.
Yeah. I'm going to show on screen here an image from David Goodsell, which I don't know if you've seen his work. He does these paintings of the inner workings of a cell. Absolutely spectacular. But in this bacteria, we can see that the DNA is all wound up. There's a bunch of different proteins that are stuck to it, and they're causing it to kink in certain ways and to bend in certain ways. And those proteins, they have an affinity to certain sequences in the genome that they attach to. And so not only does the sequence of the genome matter for coding for proteins, but it also attracts these molecules to attach to it and change how tightly bound or how loosely bound it is. And that determines how much that chunk of the DNA will actually be read and used. Yep. And so this is for gene regulation. And it's not clear that the first organisms would have had something like this. Would be something that evolves gradually over time and becomes an advantage to certain organisms over others. You had talked specifically about the if-else statement in bacteria for digesting lactose, which was the first one that we discovered. The first switch that we found, this is like right after World War II, was in bacteria.
Yeah, so it's the textbook case.
Yeah.
So this is where and it tells you so much about biology. So there are bacteria, and bacteria need a lot of food to run on, and they need it all the time because bacteria don't have big bellies or whatever. They just need
Right.
the food needs to come in right now. And they can run on different kinds of molecules. Most of them are sugar related. Like human beings, they need to have a certain concentration of these sugars in their system because otherwise they don't function.
Right.
And the same thing for human beings. And they need to regulate that. And it was sometimes biology latches on to one sort of mechanism or organism and says, we're going to study this until our eyes bleed.
Right.
And find out everything we can about this particular mechanism. And this one was picked. It's called the luck operon. You could talk about it. I made a joke that this is sort of the elementary particle of biology. It's sort of the simplest thing. We even call it an operon, which sounds a bit like a Higgs boson or whatever.
Yeah.
So we try to really understand it as a sort of the simplest unit we can find. And in the simplest way, you would say, well, if the glucose concentration becomes too low, then it starts to convert lactose into glucose. So there could be an if statement that says, if not a lot of glucose, then build lactose, or then convert lactose into glucose. And it turns out the reality, even though this is the net effect of this piece of DNA, the actual reality is like nothing like this because it's so messy. So what it actually senses is that as long as glucose is being transported into the cell, there is a sort of waste element that builds up. And the moment it's no longer entering the cell, that waste element stops building up, and the whole process actually senses a lack of that waste product. I particularly like this example because it shows you there is no intelligent designer behind this algorithm. It works. The algorithm is fine. It achieves the goal. It regulates glucose. It does that really well, but it does it in an incredibly stupid way.
Yeah.
I cannot argue with the success. It's massively successful. It might even have advantages, but it's not the intuitive thing that anyone would ever do. We would, as human beings, if you want to regulate glucose, we would build a glucose sensor and then regulate glucose. And that's not what nature has done. Nature has made this sensor that actually measures this other stuff. And the way it measures that is actually that's how these proteins do their work. Proteins are these smaller or larger molecules, and they can bend. They can be like this, or they can be like this, or they can ball up, or they can stretch out. In their stretched out form, they might not stick to a bit of DNA. Right. But let's say it changes, it bends, then suddenly it might stick to DNA. And the actual glucose sensor or the waste product sensor is a molecule that switches shape based on the concentration of that waste element. And the moment it switches shape, it manages to attach to DNA.
Right.
And there it will then promote a certain gene to do things. But it's even smarter than that because it makes no sense to start converting lactose into glucose if there is no lactose. So it's actually a dual if statement that says only do this thing if there's a lack of glucose and we have lactose, then you can do this thing. And I'll round it off by saying that people are really happy with this sort of elementary particle of biology called the luck operon. And by now I have found that there are complexities layered on complexities on complexities. And even this most fundamental thing we found, you could probably write a 1,000-page book literally only on this aspect.
Let's try and clarify this again. It's kind of hard without visuals here. So the part that I've studied most is the thing that senses lactose. So if it's a protein that sticks to the DNA and stops it from being used. So inside the bacteria, you've got a string of DNA which codes for an enzyme that can process lactose. And there is a protein stuck to that gene that stops it from being used. But if lactose is in the cell, it attaches to that protein, causes that protein to change its shape, and it falls off the string of DNA. And so that can allow that gene to start being used. It's a beautiful system because the gene is turned off when there's no food for it to digest. So if you were to try and think of the origin of something like this, having the machinery and constantly running it, it'd be like, let's say that you had a fast food chain and you had restaurants all over the country, and like 3 cities went vegan. You had no way to know that, and so you just kept running your burger joint in those cities, and you made no sales, and you had no feedback, and so you just kept doing it. You could probably survive if your chain was big enough by just losing these stores and running them at a loss. And that's what a bacteria that would constantly make this lactose enzyme would be doing. They'd be constantly running at a loss. They'd be making this protein for no reason. It would be produced. It would spend up a bunch of energy being produced. It would bounce around in the cell, then it would get digested eventually and recycled.
Well, actually, it's even worse. Bacteria live in constant competition.
Right.
So because exponential growth is the name of their game. And the only one that survives is the one that grows quickest. And the moment, and because bacteria can double themselves in like 20 minutes or something like that. And let's say you reduce your doubling time to 19 minutes because you're just slightly more efficient at using energy. After a day, you dominate everything because you've made a few more copies during the day than the rest. So bacteria that are less than perfectly efficient, they lose.
They would be winning before that enzyme evolved, but as soon as that enzyme evolved, if you don't have that enzyme, you lose. So
You in biology experiments with bacteria, and they're like really fun to do because biology is really messy, and it's not fun to do. If you know any biologist doing experiments with zebrafish or fruit flies or whatever, these are truly heroes. Because these are experiments that mess with you. But bacteria is very simple. And a very typical experiment is that you take a Petri dish, which is like a piece of glass, and you put only one kind of food in there for bacteria, and which is the food that they don't really like. And then you inoculate that with one kind of bacteria, you put it in the dish, and they start growing slowly because they're feeding on pure lactose, which they don't like. Right. So the bacteria don't grow that quickly. And then within a really short time frame, and it can happen in one day, one of your bacteria discovers how to more efficiently break down the food you gave them. And that one literally launches a new plume of bacteria that grows far faster than all the other ones. So you can literally see them figure out, okay, you gave us the wrong food and we've evolved our way out of it, and boom, now we grow 5 times quicker and we end up dominating the entire plate.
Yeah. And the really cool thing about that is that you can actually go through and find the mutation that caused that change in many cases. Yeah.
No, it's textbook. And I think it used to be that people were like, is evolution real? Well, bacteria people, they say, evolution is very real. And by now it is so real that it messes with your experiments. Right. So if you're trying to learn something about bacteria, what biologists now do, they do their experiment with the bacteria and then immediately put them in the freezer again. Right. To just stop them from evolving. So into the minus 80 they go. And then when we do another experiment, we briefly take them out of the minus 80 Celsius so that they can do another experiment, because otherwise they will out-evolve your experiment. They will no longer, you will not learn anything. You will learn that evolution is a thing.
So you have some slides here, and I'd like to just let you go through that uninterrupted.
So this is briefly the luck operon that we discussed before, which is this if statement. And what it will do is it says if there is glucose in the cell, then use the glucose in the cell. If there is no glucose in the cell, and if there is lactose, then start converting the lactose into glucose. And if there is no glucose and no lactose, then do nothing. Right. Bacteria are quite good at doing nothing in case they have no energy. And that's the third option. So on the lower right of the screen, you will see typically how a computer programmer would represent these 3 choices. And on the top, you see the luck operon, which is the nature implementation of this algorithm. And you see on the left, you see a thing called promotes RNA polymerase binding. And then you see CAP. And CAP is a sort of side product that we mentioned that sort of tells the cell that, hey, we're running out of glucose. Yeah. And if that one is present, it will latch onto the DNA. So that bar you see that says CAP site and promoter and operator, that is actually a piece of DNA. Yeah. And the moment it needs to become active, this CAP thing attaches to the DNA and it attracts the RNA polymerase, which says, I'm going to read this bit of DNA now.
Right.
So that's one part of it. It said, do this stuff. If there is a lack of glucose, then try to read this DNA.
Right.
And then more on the right, you see a thing called a repressor. Mm-hmm. And the repressor makes sure that this only works as long as the repressor is attached to the DNA. The cell can physically not read it.
Right.
Then it's blocked. The whole processing thing is blocked. So there's a promoter at the beginning that says, yes, please. And then there is a repressor that says no. And this combination, if there is actually no lactose present, then the repressor attaches to the DNA.
Right.
So, and the net effect of these two is that if there is a lack of glucose, then the system wants to read the DNA, but that will fail unless there is lactose present.
Right.
Because if there is no lactose, then the repressor attaches and nothing happens. So this is this is as close as you're going to come to actually seeing the equivalent of computer code with if statements expressed in biology.
Right. This is really amazing. So just again to clarify this, the bar here that says it starts with saying cap site, that represents the DNA. If there's plenty of sugar, then this whole thing, nothing is attached to it. Well, I guess we would have a repressor attached. If there's plenty of sugar and no glucose, and so this gene would be turned off. Nothing, the RNA polymerase, which needs to hit that gene and convert it into RNA so that RNA can then make protein, that will not happen. So as long as there's good glucose in the cell, this doesn't get used at all. If there's no glucose, then that cap gets added on there, allowing RNA polymerase to start trying to do its job to make RNA and then make protein. But if there's not lactose, that repressor is going to be attached there, stopping it from doing its job. And only when both of those conditions are met will it make this. That is absolutely beautiful. And the interesting thing is that we have all cells use the same DNA, but this if-else statement, it evolved in bacteria, and it's its own thing. And we have a bunch of these in human cells as well, but they have different evolutionary histories. They function differently, and like scientists have to discover how each of these things work independently because a lot of them are completely different. But we have these if-else statements all throughout biology.
And there are two interesting things, and there are like 1,000 interesting things here, but actually nature does a little bit better than the simple if statement, because it's an if statement that also carries a sense of urgency.
Right.
So the bigger the lack of glucose, the more intense the DNA transcription will be. Right. And so this is like an if statement that not only says if glucose is present, then use glucose, but it will also say, look, we're like really out of glucose, so get on with it.
Yeah.
It modulates it. And you mentioned the human equivalent, and I also find that part fascinating. Bacteria are within our realm of understanding. We can sit down and study this and read it, and actually you could print this bit of DNA out and you could study it and you could annotate it with a pencil and really get it.
Yeah.
Plant DNA and fungi DNA and human DNA is like 3 orders of magnitude more complicated.
Right.
So instead of only having one operon, there is a layer on top of that cascades that says now this subsystem is active or not. And that is when you study that, it is possible to study it, and we do make progress there. That's how we develop medicines and other stuff. But there is like bacteria. Then you go back to bacteria and you go like, hey, yeah, I can just understand this stuff with pen and paper.
Yeah.
And the final thing I find utterly fascinating about this, so you have this stretch of DNA with the cap site and the operator, and then there are these genes, the LAC Z and LAC Y, and they are actually bunched together. So these are a bunch of genes that have independent functions, but they're all bunched together here so they can get turned on and turned off in unison.
Right, together. Yeah.
So this is like where you have a bunch of documents and you put them all in one directory or all in one folder because they make sense together.
Right.
And that's you can recognize that. You would like a computer programmer would also put a bunch of functions in one file because they're related and it's sort of easier to keep them together. Sometimes you spot in DNA this sort of ordering where things are sort of that actually sort of do something with each other, are also co-located with each other. Which does not specifically have to be the case. You could have two genes that are intimately involved with each other, and they could be on opposite ends of the DNA or random places. But sometimes you see them grouped together where you realize, apparently there is some benefit of having these together. And here the benefit is, of course, obvious, because otherwise they would not get turned on in unison. But in other cases, it almost feels like, we have to find the reason why. There's a phenomenon called genetic hitchhiking, which can explain these things. But it ends up in a sort of very human, familiar organizational structure.
Yeah. Maybe it would be good to talk a little bit about how genes can actually move around within a genome.
They do.
This happens quite often. It's a common form of mutation, and so finding a good neighbor is almost as important as the actual sequence of the gene. So that does happen over evolutionary time.
Yeah, it does. And to the point that the vast majority of human and plant DNA consists of elements that have been copying themselves for no good reason.
Right.
Which is, and if you ever taken a look at the code of a big computer project, I mean, if you ever download a computer program or a game and you wonder why is this game 25 gigabytes?
Right.
What happened there? That's because if you would study it, you would find that you're downloading a lot of copies of the same thing that never got removed from the project.
Yeah.
And welcome to biology. That is exactly what happened in biology, where every gene has a few dying copies lying around or failed experiments, and they get cleaned up so slowly that we carry with us the entire organizational history and trauma of like 1 billion years of evolution.
Yeah. So people have probably heard the term junk DNA, which is falling out of fashion because we're finding so much function in the supposed junk DNA. But you've got pseudogenes, which are genes that should code for protein but are broke, and organisms will still have them. Humans have a bunch of sent pseudogenes. So our nose is super weak compared to other primates because a bunch of our smelling genes are broken, so they're now pseudogenes. And evolution hasn't cleaned them up yet, maybe because there's something else in them that's still important for folding up the DNA and so on. Maybe evolution can't get rid of them because there's something beneficial in them still. And you see that everywhere. And then you have these jumping genes, which are these stretches of DNA that can just make copies of themselves, pull themselves out of where they're located, move themselves. Sometimes they move a chunk of their neighbor as well. And our genome is loaded with those, and we think that those were important for organizing our genome. And they can cause chaos sometimes when they get out of hand. And it's such a mess. It's such a mess.
Yeah, it is a mess, but it is an inspirational mess.
Yeah.
So it's a lot like, if you again go back to computer programmers, we will often also not really, let's say there's some functionality in code that we no longer need right now.
Right.
But we might feel that but maybe one day, maybe we don't need it now, so let's keep that file around. But even though we don't use it,
right.
So the file is still there, and because maybe one day we feel like we could use it again, and then every software project is full of this stuff. But then one day you realize you need that code again, and hey, it's still there.
Right.
And then you can revive it, and then you find even in programming, and this phenomenon is very real and puzzling. Let's say you took a bit of code and you didn't use it for 2 years, but in theory, nothing should have happened to that code in 2 years' time. If you try to use it again, there's a phenomenon called bit rot. And that's the very real phenomenon where you take this 2-year-old piece of code, nothing changed to it, still it no longer works.
Is that because the rest of the system changed?
No, the surroundings have changed. So the piece of code turned out, so the code did not change, but let's say it relied on something. It relied on files always being given to the system in the same order, or whatever. Yeah. There's a hidden assumption. And over time, that assumption has been violated, and the files now get presented in a different order, which should make no difference, but your code was relying on some files coming first. So after a few years, your code, you revive it, you start using it again, and nothing works. And then you're like, hey, has this code been mutating while I wasn't looking? That's what it feels like. It feels like there was a fungus growing on your code, but it's actually bit rotting. And the same phenomenon happens in biology as well, that if you see these old copies of genes that are lying around, we can study them because so here's another dark secret, and this really fascinates me. In bacteria, you can see where a gene begins and ends very well, because there's a marker that says the gene begins here and it ends here. And it's even more luxurious than that. You get a warning that says the gene is about to end. And that's because the polymerase has to get off again. It needs an exit ramp. So at the end of a bacterial gene, you see the little exit ramp, and then you see the actual end. So it's very easy in a bacterium to see where a gene begins and ends. Very much like in a computer program, when we look at the ones and zeros, we can find the beginning of a function and the end of a function because it has a certain structure to it. In more advanced or complicated organisms like plants and us, the beginning and ending of a gene is we cannot find it. We really have no idea where a gene begins and ends. Nature has a fantastic way of finding out where a gene begins and ends, and it's perfect at it. And we haven't figured out how nature does that. The only way we can figure it out is by looking what nature does and then saying, well, apparently the gene begins here. When we do that, we get a list of, okay, these are the active genes. These are the ones that actually work.
Right.
And for every gene, we find like 25 copies lying around of older versions of the gene, miscopied things. And that allows you to do a whole analysis to say, okay, apparently this gene is an old copy of the other one. How much has changed?
Right.
And that allows you sort of a complete forensic historical analysis of how a gene has evolved over the years. So it's like having this gigantic archive where every email you ever read is still there.
Yeah.
And all the drafts are also there.
Yeah. You're saying that sometimes when you turn code back on, it breaks. It doesn't work right. We have atavisms in evolutionary biology. So you've got sometimes people will be born with a tail, or a horse will have horses only have one toe. Sometimes they'll have three toes, and it's always a monstrous deformity because you've got this thing that got turned back on again, but everything else around it changed. And so now you just got this weird, it's usually a disability that ends up happening.
But it could go the other way around. You mentioned the primates, that compared to primates, our sense of smell is so much weaker, which I didn't know. I find it fascinating. But let's say we had a pressing evolutionary need to get back our superior sense of smell. Mm-hmm. Evolution would quite quickly sort of rediscover these defunct genes lying around and maybe accidentally enable one and create some quite miserable people that now suddenly have a tremendously good sense of smell, which they didn't want. I know, but you find that these old genes, they're sort of there for the reactivating.
They are actually going to mutate over time, the longer something's going to be off. So it's definitely easier for evolution to turn something on, and then mutations will edit it and start to make it work with the current system over time than it would be to invent a whole new gene, right?
Yeah. And actually, we use this to figure out if a gene is important, then defects in it will get fixed, or they will not spread.
Right.
If a gene is not being used, it will just accumulate errors at a known rate.
Right.
And from that, we can even determine how long ago, how many millions of years ago, or tens of millions of years, a gene became defunct, because we know that there's now 12%. There's now 12% error rate in there, and we know from experience that it takes 100 million years for that many errors to crop up.
Yeah, and there are a lot of experiments. I'll probably try and link to some papers here where people will they'll break a gene and then see how long it takes evolution to fix it and get it back up and running, and it's really interesting to watch this happen, because you can break it in the lab using lab techniques, and then you watch slowly, mutation by mutation, you watch the cell try to fix it. Sometimes it'll just completely throw it away. It never finds a way to fix it again because it's a stochastic process. It's random, and then sometimes.
Let me tell you something that I only learned recently. Mm-hmm. Let's say you disable an important looking gene.
Yeah.
This gene like super important, and I'm just gonna ruin it. And quite often, nothing happens. And because it turns out that the work of that gene was not done by that gene alone. There were two other genes that were also doing something similar, but together.
Right.
So we sometimes fall into this trap where we think that this gene is like the gene for.
Right. Right.
And it turns out that it's not true in two ways. For one, it's not the only gene for. And so like the gene for blue eyes, maybe I'm just making this up. This is not factual, but it's if you then look it up, you find out that the gene for blue eyes actually does three other things as well. And there are also two other genes that together also make blue eyes.
Right.
And that is in stark contrast to how we think that these things work. That you have the gene that gives you X or whatever. And there is actually sort of analogy also in the computer world. Computer programs tend to be far more fragile. So if you take out one function, then your whole computer stops working. It's quite rare for a computer program to say, well, I'll limp on. Sometimes you could, for example, have multiple computer programs that have the same effect. One of them defragments your files and the other one cleans up old files. And maybe together they both have a defragmenting effect. As an example, that's more of the analogy you should be looking at.
Yeah, so I work with a lab in Corvallis that does gene knockout experiments. Well, they designed a molecule that people use for gene knockout experiments. And they found that these are using zebrafish. They found that for the most part, if you knock out a gene in sperm or egg, it sometimes won't even have an effect.
Because very often.
Other genes will compensate for that loss. But if you knock out a gene as it's developing, then it will break the embryo. So there's a bunch of just backup systems.
And we keep on discovering. I mean, when I'm in a philosophical mood, I sometimes wonder if there is not so much complexity in there that we'll never discover all of it. So we know, for example, that in animal genomes during the developmental phase, as you noted, there's actually sort of QA run that happens on DNA, and where whole genes get reactivated because the other one is not active.
Right.
So there is a sort of trial run going on during the embryo phase, where defunctive genes are sort of papered over and say, well, not you, please.
Yeah.
And other ones can then get reactivated or become more important. And that's why I sometimes think that our own brains may not be capable of understanding the full complexity of our own genome. Much in the way that a computer may also not reason about its own existence. But the bacteria are definitely within our sights. We could definitely understand bacteria.
I like to say that trying to understand our own biology is kind of like a dog trying to swallow itself. That last bite is just too hard.
No, there is no guarantee that sort of that a brain will be able to understand its own genetic makeup.
Right.
It doesn't have to be that way.
Right.
And since it's like this 4 billion year old software project, there is an ample room for complexity to creep in.
I was talking to a geneticist who studies dog morphology. And the way that he studies it is he looks for a trait, and then he looks for all the dogs that have that trait, and he sequences their genomes, and he looks for similarities and differences, and then he uses statistics to pinpoint, I think this is the mutation. And then they can genetically engineer and put that mutation in a dog and see if it's true. It's trial and error. We are blind. It's so complicated.
We are very, very much so. But we are making progress there, because it used to be, the idea was that every trait would correspond to one place in DNA. And then we found out that it wasn't true. But in the meantime, 1 million papers have been written about how this and this genetic defect gives you that and that illness. And it turned out to mostly not be true. But now we have the statistical prowess to figure out that say, look, if you have this genetic change over here and that one over there and that one over there, then together that likely means that you have blue eyes or whatever.
Right.
And that is now statistically becoming feasible. And it never used to be feasible because just the computational stuff you need to calculate that. It is a lot of work. And that will actually finally make DNA more useful, because right now, actually, when the human genome was finished, and it's actually now almost finished. It has been almost finished for the past 20 years, but we're now really getting there. And that has not been the boon that people thought it would be. We have not really been able to say, okay, you have this DNA and you should do that. And maybe I think we're now getting somewhat closer to being able to do that because we understand the interactions between mutations on different genes and different chromosomes now.
Hopefully, someone doesn't use it for evil first.
Yeah,
It's
well,
too easy to make a disease.
that would turn this into a very different episode. But I'm not happy. I am not confident that this will not go wrong.
Yeah, it's terrifying that you can actually order a stretch of any DNA online and it'll get mailed to you. So,
Yeah.
if you know the code and we've got pathogens, their DNA is up on the internet. What else do we have here in slides?
Yeah, we discussed this a little bit. So what is in our DNA actually?
Yeah.
And it's tremendous fun to do this. And we should also do this for computer programs. Because like I said, whenever you update a computer game and it's downloading 20 gigabytes of stuff, you may wonder, what am I downloading here?
Yeah.
And when you do the analysis on the human genome, although this works just as well on dogs and cats and whatever, you see the little piece of the pie in red, the protein coding genes. So those are the action bits, the bits where we say, look, these are really pieces of DNA that we see end up in proteins. And it used to be, like you said, rather common to tell the rest of DNA is just junk. And partially we know that it's not true, but from a very significant part, we do know that this was not put in here for our direct benefit.
Right.
So, for example, these LTR retrotransposons, those are just copy-happy bits of DNA that were shaped in a way that when the DNA gets copied, sometimes you get two of them.
Right.
And that leads to a disaster because that leads to a gene that gets evolved to make even more copies of itself.
Yeah.
Because if it succeeds in shaping itself so that instead of one copy you get two copies, well, next time you get four.
Yeah.
So there is massive evolutionary pressure on genes to make themselves copy, more than 100%. And from the history of various genomes, we see that in some animals that this has really led to disasters.
Yeah.
And
it's a parasitic stretch of DNA, essentially.
Yes, we don't need it. There's no, but there is a theory, however, that says that since almost no organisms have excised this unnecessary DNA, it probably has some benefits. And so one of my own pet theories, and I hasten to say that it's just a theory and a hypothesis, is that let's say you take DNA and 97% of that DNA actually does not lead to proteins.
Right.
And then you are a virus. And the holy grail for every virus is to infect our germline.
Right.
To become part of our future. And you now, as a virus, have a 97% chance of infecting a random bit of DNA that will never go anywhere.
Right.
And that is, in a way, a sort of defensive structure. Mm-hmm. Where you say, well, you can attack my DNA, but chances are you won't hit anything.
Yeah.
Whereas if you infect a bacterium, bacteria are like 90% of their DNA is actually active.
Yeah.
So whatever piece of the bacterial DNA you hit, you are in there forever. So it might be that it's like when you have a secret to hide, that you hide it in a room with lots of other papers, and you go like, well, good luck finding my secret.
Yeah.
It's here in this paper mess.
Unfortunately, HIV has a trick for that. It attaches itself to I think it's RNA polymerase, and it's got an enzyme that attaches it to that and then inserts itself right in front of the polymerase. So it makes sure to always insert itself in an active part of your genome.
Yeah. Well done.
Yeah. the viruses have hacked that defense.
Yeah.
If that is a defense, some of them have found a way to hack it.
Yeah, and I think actually it is a defense because, as I said, we struggle as human beings. We struggle mightily when we look at human DNA to find where a gene begins and ends. Even with supercomputers, we struggle. We don't, and I interpret that as not an accident, but as a way for genes to hide themselves and say, hey, you cannot find me.
Yeah. Yeah.
because we're cleverly hidden some way. But I know this is just my pet
Yeah.
theory. Um, Maybe I'm right. The introns slice of the pie, so that's the 26%.
Yep.
It's interesting to look at it. If you look at a gene that is active, it has parts that end up in proteins. And then in the middle of a gene, there are whole stretches, and they're like far larger than the actual gene.
Yep.
There are whole stretches that say, well, you should read this DNA and you should copy this DNA into RNA. You should do everything with it except turn it into proteins.
Right.
Um, so these introns, they sit there, they faithfully get turned into RNA. That RNA gets destroyed again, and there is so much of that stuff going on that we are starting to understand maybe what it's for, what it's not for. But one very interesting thing is that, and this also happens in computers actually. Sometimes in computers you need to delay something.
Mm-hmm.
So something should not happen 1,000 times or 1,000,000 times per second. It should only happen at a slower rate. And one of these ways of achieving that is making a computer count from 1 to 10 million.
Really?
Because it will take some time to do that, and that means that the thing that is happening will not happen as much because it spends time counting to 1 million every time. And we have one gene that's the longest gene in the human body, which is associated with dystrophy, and that gene is like 99% introns. It's like a million pieces of genetic letters sprinkled all over that protein, and that means that making that single protein, making that one molecule, takes like half a day.
Right.
Simply because it's so much work. And the average cell will actually also literally have one copy of that molecule, and it will never run away because making it takes half a day.
Yeah.
And that may be the crude way in which nature has said, well, look, this is a very unpopular molecule, and we don't ever want you to make a lot of it. That's why we stuffed it full of these introns that you have to read to get to it. And then it fills your whole cell with bits of RNA that are not used, which again probably has a reason, but it's not something we've figured out yet.
Yeah. It's really interesting to see how much inside the cell gets built and then immediately broken down again. I mean, things are getting digested as they're getting built in some cases. It's really
no. There is a whole metabolism going on there, and that metabolism is very rapid and efficient, and make sure that the cell is. I mean, the other thing to realize is that cells will happily, at least in our body, sacrifice themselves. They have there's no pride attached to that because you can make new cells. And it took me a little while to realize that, because the cell in a mammal is such a cathedral of complexity and capability that this sacrificing one, it feels like a waste of how could you?
Yeah.
We built this thing. And but then of course with the whole coronavirus thing, I started trying to understand the human immune system. And it turns out that we just kill cells if they look the wrong way.
Yeah.
When we're not sure about the cell, the human body just kills it. Says, "Well, we can make other ones."
Yeah.
But otherwise, the efficiency is just astounding. Every part is used for something.
Yeah. It is really fascinating. I mean, just to think that people make babies on accident all the time, and it is the most complicated thing ever. But we just do it on accident because we're so.
I mean, sometimes I just sit. So, like I said, sometimes I just sit there and I'm maybe eating a banana, and I'm like, "Wow, all this glory," and the whole info. I mean, if you realize that a cell has these vesicles and these factories, and so the cell makes a protein, but the protein still has to go somewhere. So there is an address label attached to it, which is part of which is then used to get the insulin molecule to the right place, where the address label is stripped off, and then the cell gets further assembled, and you're like, oh man, this is just
it is amazing. And the complicated chemistry and engineering that evolution did to allow you to pick your nose is just spectacular.
And it's not. I mean, biologists, actual biologists, because I'm just a pretend biologist, but they you alluded to this earlier, that there is this magic sense of awe,
Mm-hmm.
where we go like, look, this stuff is like so amazing.
Yeah.
And then in the next sentence we have to say, yeah, but it's all fully. I mean, it's completely logical. It's science, and we'll figure this out. And it's fully understood. We fully understand how all of this works, because we have to say that. Because the moment we say that we are in awe of this miracle of biology, then there is a whole group of people that go like, you admit it's a miracle. And it's tricky to strike that balance. And no, we know we can understand all of it. We have the blueprints. We can understand it. It just requires work to understand it. But at the same time, we are just super in awe of this magical technology, which is not magical at all. It's just like Arthur C. Clarke said, that any advanced form of technology is indistinguishable of magic.
Right.
And until you understand it, then you go like, okay, I get it now. I get what's going on, and I'm still in awe.
Right.
Of it.
And this is the thing that has kind of bothered me with the online atheist community, is that people are wanting to. It seems like they're wanting to dumb things down and make it. Oh, things aren't that fascinating. They're not that special. But no, like, I mean,
They are.
The thing that gave rise to your mind and your ability to think and behave in this, of course it's spectacular. It has to be the most spectacular thing that you'll ever study. It's really unfortunate that people give in to this idea that if you let yourself be in awe, you're opening up the door for woo-woo. Yeah. And I don't think that has to be the case at all.
No, and I am here in Europe. I have the good news is it's not as polarized here.
Right.
But even here, the moment I would go out and say, look, we don't, so that there are a few biologists that just go out and dare say it and say, look, yeah, we don't know where all this came from. Yeah. We're searching for it, and we see the process. The process is logical, but there are still steps where we really don't know. And it's important for scientists to be able to admit that, and to even say, look, and the some of the things that we will find will be awesome, and hard to believe. And the risk is that the moment you admit that, they say, aha, evolution has failed. Whereas evolution is, like I said, so real that you see it happening during your experiment, even when you're not set out to prove evolution. Well, evolution proves it to you. It's there. And yet, if you look at the whole set of DNA, it is so awesome that at this stage you cannot say that we understand exactly where it came from, and that we can explain every bit of it. That is not true by a long shot.
Hopefully I will have some people who are experts in the ribosome on eventually. But the origin of the ribosomes and the translation system, we have
that is where the magic happens. And to me, I mean, if you would tell me, look, we just sent yesterday humanity sent a new robot to Mars to do investigations, and if someone puts a DNA sequencer on Mars and turns it on and finds DNA, I would not be surprised.
You're saying DNA from an original origin?
Yeah, from just where we would say, look, DNA. Maybe we did not invent DNA here. Maybe we didn't. Maybe it existed already.
Oh, okay, yeah.
I'm not saying that it has to be the case, but let's say we send a DNA sequencer to Mars, and it finds DNA there. Then we're like, yeah, well, I'm not surprised. I'm saying this because it will probably happen within our lifetimes. Yeah. That we send the DNA sequencer to Mars. So it's always good that if you say I'm not surprised, it's always good to say that before it happened. Because otherwise it doesn't count. Yeah. But there is a black hole there. We see the glory. We see, like I said, we see the sort of vestigial patterns that the codon used to be maybe 2 DNA letters. Mm-hmm. We see hints of that, which would create a simpler ribosome. We can imagine a ribosome that exists solely of RNA.
Right.
So we actually have a bootstrapping mechanism, because we know right now you need a ribosome to make a ribosome.
Right.
Which is not a very... but we know that you could bootstrap a ribosome using only RNA.
Yep.
We can still see that. We can see that in the core of the ribosome is an RNA-only construct.
Yep.
And there is evidence that it would work if you would take out all the proteins. It would just be very slow. It would still work.
Yeah.
So we actually have an evolutionary path how you would get to a functioning ribosome. It's just that, we haven't seen it.
Right. Right. And we don't have a clear idea of how RNA would have RNA chains would have been produced and evolved without enzymes, because right now all of our RNA evolution experiments, they require enzymes. So that's one of the big questions in Origin of Life is, was RNA
But
really first?
I see people obsessing over that they say, we haven't found this, we haven't found that. Yeah, but we also have not found that, we will not find it.
Yeah, right.
It's a matter of imagination. There are no impossibilities in there. It's just what is the path and how would it happen, and what are the conditions. So the fact that we don't have an open and shut case is not super relevant. And I am more intrigued by the fact that we see that within this ribosome there is this core of RNA that could build a ribosome.
Yeah.
Because that tells you that that's no accident. That it was that way, and that once the ribosome existed, it used itself to make itself more efficient. Stands to reason. So the gene, people, when they looked at it, even before computer programmers, people assumed that the gene was some kind of sort of functional unit,
right?
And like a program or a function or whatever. And because we were human beings, the gene was known before the whole DNA was sequenced. And people said, look, because we're human beings, we must have 1 million genes. So even biologists can be quite irrational on that front. So, and then we started measuring it, like, well, maybe we have 100,000 genes. And then later we found out that we actually have less genes than a potato.
That's great.
And that took a long while for people to realize that what you think of in terms as complexity, it's not like if you have 1 million genes that you would be a more capable animal or a more capable organism or whatever.
Yeah.
And this is a sort of the same thing that if you look at the computer program and it's like really big, it doesn't mean that it's really good.
Right.
And typically it actually means that it's not. And what I find also very exciting is that we have these 20,000 or 28,000 genes now. And if you look at big organisms, there are only a few of them that have done a house cleaning exercise. And one of them is a very, very modest plant, and it floats on water, and it's the most compact genome that we know about. And that's a whole plant, and it functions on like 5% of the DNA that we have.
Really?
And much like computer programmers are always trying to find pieces of code that are sort of minimal but complete, because we enjoy that. We enjoy having this tremendous concentration of functionality without all the bloat. And in the same way, we have these small plants that offer sort of a whole organism, but then without the 97% junk around it. And it doesn't mean it's junk, but it apparently means that you can get a whole plant out of it that functions. And
do you remember the species?
Yeah, I can email it to you. I keep forgetting it. So because we have, on the other hand, we have it's so glorious. The amount of DNA. So here on the left, there's this plant, the Paris japonica plant, which it's a pretty plant. I mean, don't get me wrong. It has 10 times more DNA than we do.
Wow.
But it's a lovely plant. I have to admit that. But you could look at it and I tried to get one of these plants for one of my presentations, but it's not biologically an interesting plant, and it's also not very pretty. And people, it doesn't survive very well in your house, so it's very tricky to get one. But it's a pretty plant, but it somehow carries along 10 times more DNA than we have. And then there's this fish on the right, the marbled lungfish, which also has around 10 times more DNA than we do.
It's a species, I think it's a species of mouse that has 4 pairs of chromosomes instead of 2. So these weird doublings happen throughout
Yeah.
evolutionary history.
It turns out birds also mess a lot with that stuff, and they have different rearrangements going on. And that's also one of the stranger things. If you really delve into DNA, then you find that human DNA is extremely well studied.
Yeah.
For obvious reasons. And then there are a few bacteria that are super well studied, and the zebrafish that you mentioned as well. We know. I mean, if you care about your health, it's a very good idea to be a zebrafish.
Yeah. Yep.
Because we know really everything about zebrafishes. And then you ask. So I recently got fascinated by birds, and I don't know, I must be getting old or something like that. And then you just try reading up, what do we know about bird genomes, and you go like, yeah, they have genes.
Yeah. Yeah.
Because like crows are utterly fascinating animals, but we know like 25 times more about chicken DNA.
Right.
Because it's a big business, and no one makes any money from crows.
Yeah.
So there are a lot of history, there are a lot of mysteries, and that's one of the reasons also why I promote this whole DNA stuff, that it could use a lot more attention.
Right.
And because of the way the world is structured with open data and all the processing power that people now have at home, people that are good with data systems could really, for example, improve the state of the chicken genome or the crow genome. And that's not the chicken genome, because there's a lot of commercial effort focused on that. But if you and I would say, hey, we're going to study the crow genome, we could download the stuff and we could create a scientifically relevant paper in 2 months.
Really?
And it would further the state of the understanding of crows, simply because right now not that many people are looking at them. But you don't need to be this. You don't need this 20-year track record of biology in order to advance the state of understanding of crow DNA.
You had an animation of the ribosome doing its thing that was really cool.
Yeah, it is. I think it's this one, but it's...
Yeah. So here we see the ribosome. That's that little black chain going through it. That's the messenger RNA. Coming into it are the transfer RNAs bringing amino acids to the ribosome, and those transfer RNAs are attracted to the codons in the messenger RNA. So every 3 letters of the messenger RNA attracts a specific transfer RNA, and you're seeing the new protein that's being built is just starting to peek out the top there. And what is that, like black? That's the new protein. Now it's being attached to a cell membrane so that it can excrete that protein into a chamber that's being built. But yeah, that ribosome, the core of the ribosome, is identical in all organisms. A lot of the outer bits of it are unique to, bacteria versus archaea, eukaryotes. There's a lot of unique parts, but the core is identical, which is quite spectacular.
It is. I mean, and if you want to focus on sort of the magic, this is definitely where the magic happens.
Yeah.
And even in one sense, it's very identical, but in the other sense, there are these small changes. Mm-hmm. So, for example, in our mitochondria, which are the little bacteria that live within our cells to provide us with energy. So, thank you, bacteria. They run their own ribosome, and they have their own transfer RNA. And it's all very minimal, because the way it evolved is that everything that we could share with that little tame bacterium was actually taken out of it and is part of the nucleus of our normal DNA. Mm-hmm. So the only thing that is left in the mitochondrial structure is stuff that it really cannot do without.
Yeah.
And so there you find a really minimal system. And what is really fun is that even if you take everything away, you cannot take away the transfer RNA. Because if you have no transfer RNA, you're not an organism, you're not doing anything. So if you look at the tiny DNA of the mitochondria, that consists mostly of 23 transfer RNAs, because it has to have one for every amino acid.
Right.
And that's a functionality that it could not do away with. So the DNA of the mitochondria really consists mostly of the table stakes of how can I be a minimal organism.
Yeah, yeah.
And then there are a few mitochondrial bits left.
I'm curious how important Claude Shannon's work has been to the field of programming. Actually, I've gotten mixed messages from people. Some people say, well, what he did was mostly for engineering. And other people are saying, no, what he did was he really described what a communication system is, and that helped us kind of pull away from communication systems as we knew already. We'd already invented telephones and so on. And we realized we could build the communication system out of anything. And so the computer is just a diverse collection of different communication systems and different ways to translate languages from one system into another. And yeah, I'm curious what your thoughts are on Claude Shannon's work.
Yeah, so the people that doubt the importance to his field are probably blinded by how humongous it was what he did for the field.
Yeah.
So some things are so big that you cannot see. It's like standing next to, if you stand on a big mountain, you might not realize that you're standing on the big mountain.
Right.
If you look at the two most important papers that Shannon wrote, the first one was where he said, look, we have this Boolean logic thing, and you can turn that into circuits, electrical circuits, and you can perform logic with electrons.
Yeah.
And that is the fundament of everything that we do.
Yeah, yeah.
It is, like I said, it is so big that you might not see it. And would someone else have chanced on that eventually? Of course, someone else would have chanced on that eventually.
Yeah.
But his paper, and which is, by the way, he graduated on that. It's his thesis.
Yeah.
Defined the whole field and saved us like 20 years.
Wow.
Because he not merely raised the possibility. I mean, that's the awesome thing about Shannon. He did not merely raise. A typical scientific paper will go like, "Hey, well, we figured this out, and it might. This is what it works. It might have the following uses."
Right.
And that's where it ends. And the typical Shannon paper, and I read a few of them, reaches that point where it says, "Well, it might have a few uses," and then you think the article will end here, and then it's when it gets going.
Yeah, yeah.
then it goes like, well, let me define for you exactly how all that is going to work, and the speeds you could achieve, and how the topology of that whole thing would be. So I think that you could say that if you look at that first paper, the Boolean logic with integrated circuits, that not only proves that it's possible, but then tells you how to do it, which in itself would eventually have happened, but maybe 10 years later, or maybe 20 years later, or not as well. In any case, so that's the first paper. That is maybe not so relevant for biology because it's not about signals. The second one, A Theory of Communications, does the same thing. It starts out with if you read it today, that paper, you will read it and then you will think, yeah, this is a good introduction to the field. This is nice, thank you.
Right.
And then you're like, oh wait, this defined the whole field. This before this was written, it's not an introduction. It is the genesis of the field.
Yeah.
And then I'll just give you, if you remember, when you hit the end of the introduction, where you think, oh, this is a good introduction, then the paper gets going.
Yeah.
And it describes to you the limits of communications, and even the concepts of entropy, and where we can talk a lot about why he used the word entropy, but where he even says, look, there is a degeneracy in information that you could take out half the words of any sentence I speak and it would still make sense.
Right.
And then he describes the limits on that stuff. And that has very deep biological significance again.
Yeah.
Where you see that the many ways of writing codons still survived, but the redundancy in there means that many errors can be corrected again.
Right.
And so I think that paper really is the tour de force that actually shows you a lot of the signal and information content theory on how you can think about DNA. But this influence goes beyond that, because there's actually, in biology, if you have an ecology of all kinds of different organisms living together, and you want to know, is it a simple or a complex ecosystem, and that's actually called the Shannon complexity.
Yeah, yeah.
So the whole number, the very number we have to discuss ecologies is named after Shannon because he defined that stuff.
Yeah, yeah.
So yeah.
So people were programming before Shannon. So we had all sorts of coding systems. We were doing communication in all sorts of different ways. But it. Am I right in saying that he's the one that really helped us figure out what the heck we were doing? Because we were doing it before, but it was like you would get some mad genius who figured out a way to build this thing, and someone else who's just everyone else just copying that, and then he gave us.
The framework for it, because it's a very concrete example. When the first transatlantic cable was laid, people found out that they could indeed transmit voltages over that cable.
Right.
So you could put 5 volts on one end, and eventually on the other end also a voltage would come out.
Right.
But what they found is that in the original transatlantic cables, the cable had a gigantic electrical capacity.
Yeah.
That meant the moment you put 5 volts on it, it would take whole minutes for the 5 volts to also appear on the other end.
Yeah.
And people said there's something wrong with the cable. It's not transferring the voltage correctly, but it was transferring the voltage correctly. It's just that in terms of signal capacity, it was rather useless.
Yeah.
Because the bandwidth of the cable was like a tenth or a hundredth of a hertz.
Yeah.
And people were massively frustrated over that. They put bigger and bigger voltages on the cable to speed it up. But if you and it took like 5 years before someone realized what was going on. But anyone with even a cursory understanding of Shannon information theory would immediately have said, yeah, this is your channel.
Yeah.
This is, you will never get more than 1 word an hour over this channel.
Right.
Because that is the capacity of your channel. So yeah, before that I think before that we could only reason about infinite capacity channels.
Yeah.
Where this simply was not a factor.
Right.
And then afterwards, now you could say, well, we know how much information you can put through this system, and it's also not going to be more than this ever. So yeah, I do think that we've not. There are. I mean, the true mark of excellence also is if you invent something, and then a few decades later, people find your invention to be so fundamentally part of the world that they no longer realize that it was an invention.
Yeah.
That it's a cruel punishment, but it's a cruel punishment that there are now people on the internet that will go, yeah, sure, that makes sense. You take the logarithm of the signal to noise ratio, and then you multiply that by the bandwidth, and then you get the capacity. Obvious.
Yeah. Well,
not.
Yeah. It's kind of like when you watch 2001 Space Odyssey. It's like, this is just copying all the sci-fi that I've ever seen. Like, oh, wait, no. Everything else is copying this. Okay.
Yeah, or even worse, they made that movie before the first people went to space.
Right.
So it's literally, so it's by now you go like, well, yeah, it's a bit different than that, but good for its time. And you realize that it happened years before anyone got launched into space.
Yeah.
And but if we go back to Shannon, he actually wrote more words about biology because he was also he recognized that this was a very fascinating system. And he also did research into artificial intelligence and neurons and other things. And I think that there are people out there that cannot deal with the concept that someone is just so much smarter and greater than you are. Yeah. That you somehow have to belittle his or her achievements.
Yeah.
That and they say, it's obvious. It's obvious now. I learned that stuff in school. Yeah, you know why you learned that in school.
Yeah.
And it's the same thing happens to Einstein. People keep saying, yeah, Einstein was wrong. No, maybe he was wrong in some places, but he was a giant of his field. And similarly, we don't hold the smartest people I know today, and it's worrying, don't hold a candle. To what a guy like Shannon achieved.
Right.
And
yeah, I wonder why that is. I mean, sometimes I think, well, maybe, in biology it's Darwin who figured out. I mean,
yeah. Darwin was actually he was a very human person. If you read his diaries, one day he wrote, I hate everything and everyone. Right. I'm not gonna do anything today.
Right.
It's a very human thing. And the other thing about Darwin is that he also said, look, I'm not the smartest guy in the world.
Right.
And interestingly enough, he was not, but he was the most dedicated person in the world. And he did not let go. And he was very, very methodical.
Yeah. one of the things you see in biology, you get people that are really upset that they're working in Darwin's that they're working on the program that Darwin set up. They want to you'll see people saying, "Oh, Darwin was wrong about this," and look, evolution doesn't work how he says it does. Like there's like a whole website of people called The Third Way, where they just try and put themselves above Darwin, and it's really interesting. Like people don't want to just work in someone else's house that was built, but you have these people. In Darwin's case, I think you're right. It wasn't it's not that he was a super genius. It's that the idea came. It was time for this idea to come about. We had enough background information. This was just gonna pop out of there. But with Shannon's work, you have an absolute genius who figured this stuff out. But what he did is he opened the door so wide that even another genius like him is still just going to be working on what he started, because there's so much to do within that playground that he set up for us.
Yeah.
So
but similarly, people keep trying to prove. So there's this Shannon limit, which is to say this is the amount of information per second you will get through this medium.
Right.
And it's like the light speed.
Yeah.
You will not break the speed of light. You will not break the Shannon limit.
Right.
Still, the whole world is full of people that have found ways to sort of say, yeah, but I did break the Shannon limit.
Yeah.
And because somehow it's just a limit. It's a challenge that someone put there.
Right.
It says, look, you're not going to get beyond this, and that always makes people want to say, well, I will get beyond this.
So you're saying that's kind of like breaking the Shannon limit is like breaking the second law of thermodynamics.
Yeah,
no one
or the speed of light, or the perpetuum mobile. That gets people all riled up, and of course with the Shannon limit for a very long time was also a commercial thing, because it limited this is the amount of information you're going to ever send through this copper pair.
Right.
And if you would be able to beat that limit, you would become very rich.
Right.
And we now know that you're not going to beat that limit ever, but that did not stop a whole bunch of charlatans trying to make it happen.
So now the focus is on compression, right? So you compress your data in ways, or simplify it. I had heard, maybe you know about this too, like that Microsoft Word, nobody actually knows how it works anymore. Yeah. And there's a bunch of stuff that we think is garbage, but no one dares take anything out.
and that is exactly true. And that is, but you see the same thing in life. There are probably parts of biology that many organisms have tried to take out.
Yeah.
And no, it doesn't work. One of these, for example, is sexual procreation.
Right.
It turns out that beyond a certain complexity, it's impossible not to do it.
Right.
And there are many organisms that have tried it, like, for example, yeast. Yeast is a good example that can, it's a complex organism, but it can procreate asexually. However, occasionally it still does it. Yeah. And this turns out to be one of these mysterious things that you cannot take out and survive for a very long time.
Yeah.
And but Microsoft Word, I mean, Microsoft Word is in a sense what happens if you leave a piece of biology alone for a few years, and then you see what happened in your fridge.
Yeah.
And it became a lot bigger, and no one really knows what it originally was.
Yeah.
And, which is why bacteria are so fascinating, because bacteria have this minimal DNA. Everything about the bacterium is minimal, because otherwise it would not be there. It would not survive. And that makes them a lot more fun to study, in my eye at least, than studying the humongous complexity that is even a yeast cell.
Yeah.
A bacterium is within striking distance of being a set of algorithms.
Yeah.
And in the sense that you can also use mathematics to predict their behavior, and they will do that. They will even, there's a beautiful experiment that you can run in a Petri dish where you set up two colonies of bacteria, and they will grow, and in the middle where they meet, they form an interference pattern.
Yeah. Yeah.
And you can calculate the shape of that interference pattern, and they will dutifully do it for you.
Wow.
And so the problem is, and that's one thing, there are aspects of biology that are not present in bacteria.
What do you mean?
Well, for example, many animals are able to do navigation based on the polarization of light,
right,
or magnetic fields. So doves, for example, are able to do magnetic navigation, but we're not quite sure how they do that.
Right.
And doves are not easy to experiment on because they're rather big and they poo, and they're not nice to have in a lab. But so I tried very hard because the lab where I worked only did experiments on single-celled things. Only no doves, no fruit flies, no zebrafishes.
Right.
Only bacteria. So I hunted, is there any bacterium that is sensitive to magnetic fields? It turns out, yes, there is, but they go by it in a very biological way. They eat some iron.
Oh, really?
And so there are actual bacteria that do navigate using magnetic fields, but they do that because their little pieces of iron actually just point north.
Yeah.
Which is not the exciting thing. Like the doves, we know that when doves sense magnetic fields, they do that using complicated quantum mechanical processes that we do not understand.
Right.
We don't get how a dove does what it does.
Right.
And but one thing you could do is you could try to take that piece of we know which genetic code the dove uses for that.
Okay.
And it makes a protein called cryptochrome. And we could take the DNA from a dove, put that in a bacterium, and see if we could evolve a bacterium that is sensitive to magnetic fields.
Yeah.
That would be a lot of fun.
Yeah.
Because then you get this minimal organism that you can study and see if it is sensitive to magnetic fields, and then do all your experiments there.
Yeah. Oh, yeah. That would be amazing.
Yeah, that would be fun. But sadly I have no biological lab at home.
Yeah. And with COVID, most labs are shut down right now.
Yeah, they have better things to do right now. Yeah. And there's also a huge shortage of materials right now. So there are experiments that you simply could not do right now because the materials are all doing worthier things right now.
Yeah.
So for now, this will remain a thought experiment.
Well, Bert, this was amazing. This was a really cool conversation.
Yeah,
I hope that everyone can just realize how spectacular our bodies are. I mean, we're the inventions that we're making, we're thinking that we're inventing them for the first time, and then we find something similar somewhere in biology. I mean, even I guess maybe wheels don't exist on animals, but we do find things like wheels at the biochemical level. I mean,
we do
everything. So it is just spectacular. Don't let a fear of woo-woo stop you from being just absolutely mesmerized by the beauty that is life.
Maybe to end this on Richard Feynman, the big physicist, and he's more controversial now than he used to be, but he still had some very great insights. And he said, look, the more I know about a flower, the more I understand about what gives it its color and why it has its color and how the color works. It doesn't reduce the beauty of the flower in any way.
Right.
It only makes it prettier to me because now I know that not only is it superficially a pretty thing to look at, but underneath there is a whole separate structure of beauty, which I can now also appreciate.
Yeah.
And in that way, we can all, appreciate nature more the more we know about it.
Yeah. Yeah. Well, thank you for coming on. Stay curious, everyone.
Yeah.